In eukaryotic sexual reproduction, the meiotic bipolar spindle captures and segregates homologous chromosomes. This process relies on microtubules pulling on kinetochores, which are assembled on highly repetitive satellite DNA called centromeres and pericentromeres. As depicted here in this schematic in Part D, the pink regions are the chromosome arms. The parts in green are at the end, depicting the pericentromeres organized on the major satellite in mice. The pericentromere connects centromeres and chromosome arms, and they regulate kinetochore microtubule attachment. Both centromeres and pericentromeres are composed of highly repetitive satellite DNA, which poses challenges for chromatin packaging. In this project, we disrupted the packaging of the green part, the pericentromeric major satellite in mouse oocytes, and tested its impact on female meiotic division. To do so, we used the trim-away method, which requires three components to acutely deplete a protein of interest: the antibody that recognizes that protein, the mRNA coding for the TRIM21 ubiquitin ligase that marks protein for degradation, and the proteasome where the degradation happens. For the control, the IgG antibody is nonspecific. For the other group, the HMG1 antibody acutely depletes the HMG1 protein, which is known to package the pericentromeric chromatin in mice. We call this group the trim-away group. Here, a massive stretching of the major satellite happens, which we know depends on the forces generated by the spindle microtubules. As in the schematic below, the green regions are now no longer compact in the trim-away group. Using this method, we first tested how failure to package pericentromeres affects the organization of centromeres and kinetochores in meiosis. Using immunofluorescence, we visualized centromeres and kinetochores. We showed that both structures are present in HMG1 depleted cells. However, the HAC1 intensity decreases in the trim-away group, and the kinetochores often cluster, suggesting disrupted organization of the kinetochores. Based on the clustering effect, we also inferred that the pericentromere packaging may help withstand the meiotic spindle forces. Since kinetochores are known to promote spindle bipolarization in female meiosis, we tested the bipolar spindle assembly kinetics by counting the percentage of bipolar spindles at different times after the nuclear envelope breakdown. We found that bipolar spindle formation was delayed in the trim-away group. That delay was specific to early meiosis when pericentromeres are stretched the most. As time goes, although eventually the bipolar spindle assembles in the trim-away group, these bipolar spindles are thinner than the control, and they rarely congress chromosomes at the metaphase plate. To better understand how failure to package pericentromeres affects the early steps of bipolar spindle formation, we used a chemical inhibitor, SDLC. It inhibits the motor protein Ag5 that drives the transition from a typical monoester to a bipolar spindle. Here, the green channel is a spindle. Compared to a control, the monoesters in the trim-away group are highly eccentric, suggesting an impact of pericentromere packaging on early female meiotic spindle organization. In conclusion, our data show that HMG1 depletion leads to defects in the pericentromere packaging and meiotic spindle assembly. We propose a model that HMG1-mediated pericentromere packaging helps withstand meiotic spindle forces, which favors timely bipolar spindle assembly in female meiosis by preventing kinetochore clustering and promoting transition from monoester to bipolar spindles.